Hello, my name is John Lucky. I'm Senior Director for Marketing at HTG, and I'd like to thank you and welcome you today to our seminar given by Dr. Catherine Poe-Geil of the National Surgical Adjuvant Breast and Bowel Project, better known as NSABP, titled Extraction-Free Quantitative mRNA Screening from Low-Input FFPE Tissue Using the HTG EdgeSeq Technology. Before we get started with Dr. Poe-Geil's presentation, I'd like to take the opportunity to introduce you to the HTG EdgeSeq processor and chemistry. This combination of instrumentation and chemistry and proprietary chemistry offers an extraction-free, highly multiplexed gene expression solution directly from many sample types, including formalin-fixed paraffin-embedded tissues. The core of the HTG EdgeSeq chemistry is the nuclease protection probe, illustrated here in orange on the screen. This 50-base DNA oligonucleotide is designed to specifically hybridize to one mRNA. It is flanked on either side regardless of its gene-specific content with universal wing sequences illustrated in gray. The gray wings are then hybridized to two wingman sequences to make them double-stranded. When added to the sample lysate, the nuclease protection probe will hybridize specifically to its targeted mRNA. The probes are added in tremendous excess so that every target mRNA in the sample will hybridize to a nuclease protection probe. We then add S1 nuclease. S1 nuclease is a powerful single-strand specific nuclease which removes all the unhybridized probes as well as all the single-stranded RNA within the sample. This leaves us with a structure second from the top, which shows the protection of the orange nuclease protection probe with the mRNA sequence. We then eliminate the target RNA with heat, and we then add the appropriate barcodes and molecular adapters for the sequencing platform we use using a simple PCR reaction. At the end of this PCR reaction, the samples are then quantitated, pooled for sequencing directly into the workflow of either the Illumina or Ion Torrent sequencing systems. As previously mentioned, the HTG EdgeSeq chemistry is compatible with a variety of different NGS sequencers. The core chemistry is adapted through the use of platform-specific adapters. This leverages the NGS instrumentation already in our customers' labs and reduces the capital expenditure requirements, allowing for easy adoption of the chemistry into the laboratory. Flexible kit configurations are available to optimize sample multiplexing and to optimize sample throughput, reducing sequencing costs for each sample. The HTG EdgeSeq chemistry is designed to utilize a variety of different sample types in a no extraction format, including FFPE tissue, where as little as 1.5 millimeters of a 5-micron section total surface area is enough to obtain good results with most of our assays. Plasma and serum can be run in our microRNA assay utilizing as little as 15 microliters of input. For our mRNA and microRNA assays, packaging and cell lines can be run using minimal sample input amounts easily achievable in most research settings. If you already have extracted RNA, most of our assays can run with as little as 10 nanograms of sample input and provide excellent results. HTG offers a variety of HTG EdgeSeq panels focusing on high-value oncology targets. The HTG EdgeSeq Immuno-Oncology panel focuses on 549 immuno-oncology-related genes. The HTG EdgeSeq Oncology Biomarker takes a larger view of biology and looks at approximately 2,600 oncology-related genes. The HTG EdgeSeq Lymphoma looks at non-Hodgkin's lymphoma subtyping and analysis, while the HTG EdgeSeq microRNA whole transcriptome array looks at microRNAs expressed from a variety of sample types, including plasma and serum. And on that note, I'd like to thank you for your time and attention, and I will hand the reins over to Dr. Poe-Geil for her presentation. Thank you.
Today I'm going to talk about extraction-free quantitative mRNA expression profiling using low-input formalin-fixed paraffin-embedded tissue and using the HTG EdgeSeq technology. I'm from NSABP, stands for National Surgical Adjuvant Breast and Bowel Project. We're also part of NRG, which is consists of 3 legacy clinical trial groups, namely NSABP, Radiation Therapy Oncology Group, and Gynecology Oncology Group. As I mentioned, we're a clinical trial group. Of course, we conduct clinical trials, both phase 3, adjuvant and neoadjuvant, and the earlier phase 1B and 2, both adjuvant and neoadjuvant. We collect samples. I'm part of the NSABP pathology lab, so we use tissue or blood and develop prognostic or predictive tests. We also profile tumors or blood in smaller studies for hypothesis generation. A lot of our work has been quite challenging, although we've been fairly effective, because we use formalin-fixed, paraffin-embedded tissue. Of course, for RNA work, they tends to be quite degraded, chemical modifications, and low yields of RNA complicate these studies. And also, recently we have done tissues using biopsies. And here you have small tissue sections or small amounts of tissue. And these tissues are particularly important in these neoadjuvant and metastatic tissues that collect these biopsies, is that it allows you to look at post-treatment samples in some cases and allow you to identify molecular changes that can be seen and differences between primary and metastatic samples. So the purpose of the talk today is to demonstrate the utility of using HTG EdgeSeq technology for gene expression analysis. And here I'll show you some comparison between the EdgeSeq technology and two gold standards, mainly TACMAN and nCounter data. We'll use different amounts of tissue, and what I'm showing you here is a single section. This is a nice sized amount of tissue. But as many of you realize, this section is not all tumor. And here I show you just that part that represents tumor. And I'll also show you data in which we use the recommended amount of tissue, mainly 12.5 millimeters squared of a single 5-micron section. And we'll also use smaller amounts, specifically a half of that amount or a quarter of that amount. And we'll also use 2 times more in an effort to understand what is the limitation for this technology. So the first pilot study will have 3 sources of material that we took a look at. Cell lines, a commercial FFPE sample, which are slightly more ideal in a vendor, has control over the processing time and the amount of tissue, how old the block is. And then we'll use some routinely processed samples in which there's little control over how the tissue is processed, other than it is processed in formalin. And we have, importantly, we have TACMAN and nCounter data for that. And we'll also look at some routine biopsy samples for our information. So we'll also use for in experiment 1 or run 1, use what we had currently available in November 2015, which mainly was one-touch templating and the V3 sequencing kits, which have changed since that time in November. So this is the results from just one sample in which we used the different lysis. We had a different run on the EdgeSeq technology, and we made a different library. And this is the correlation between those 2 differently prepared, the same sample, but so this is a technical replica, 0.99. Looks quite good. Now I'll talk about experiment 2, and this will consist of 2 different runs. Now we're looking at an updated procedure, meaning that we are templating and loading with the Chef instrument and with the new kits that are appropriate for that new instrumentation, and also new sequencing kits, mainly the high-Q kits that are now available. We'll compare between platforms, between the HTG using the OBP panel, and we'll use TACMAN and nCounter. And we have 2 different code sets, custom code sets that we've used. Here I'm showing you, just to give you an idea, between platforms, even with very good gold standard technologies such as nCounter and TACMAN. Correlation between them varies in our work between TACMAN and nanostring code set 2 and 1, between 0.506 to 0.63. Between the 2 nanostring code sets, even in this particular sample, was 0.77. Now, what I'm showing you is a correlation matrix between technical replicates and between chemistries and between different amounts of tissue. So you can see in the upper right a full technical replicate between run 1 and all within what we're calling run 1, although they were run at 2 different times, is a full tumor sample using a breast cancer sample and 71.1 millimeter squared tissue. So nice size sample, and the correlation's 0.995. Now from that same sample we've used different amounts of tissue ranging between that full tumor sample at 71 down to only 3.1 millimeter squared area in a 1/5 micron section. And here we see the correlations are quite good within the same chemistries, meaning ranges between 0.956 and 0.981, and that would be represented in the lower 4 rows of correlations. The correlation matrix, the lowest one is between those 2 chemistries, mainly one touch versus the new ion shift, and here the correlation drops down to 0.894, but the full technical reference is 0.995 in the old technology. And so that's a breast cancer sample. Now I'm going to look at a colon cancer sample, and here we see again different amounts of tissue ranging from 81.8 millimeters squared, so a full section, full 1 section, up down to again 3.1 millimeters squared. And here you can just peruse this, and if you're looking within run 2 or within that 1 chemistry, the correlations are quite good for the most part. The lowest that I see on this would be between a 3 millimeter squared tissue sample and an 81, using 81, and drops down to 0.85. But in general, all these different amounts of tissue show quite good correlations, generally all above 0.95 for these different amounts of tissue, same tissue. So now I'm going to show you between platforms. One sample run with different amounts of tissue with HTG, and there you see the correlations are quite high, around above 0.95 for the most part. You see the correlations between nanostrand 1 and 2 code sets, now up for this sample about 0.83. And similarly, but between platforms, again in this sample, the correlations are about 0.5 to 0.6. And HTG, between HTG and either nanostring or TACMan, the correlations are all above 0.6. So compares favorably with the platform differences seen. In fact, it's better than what's seen between nanostring and TACMan. Here I show 4 different samples that have been run on TACMan and 2 different code sets of nanostring. And what you can see these are bar plots, and the lines represent the median or average of the correlations. And what you can see in general that even within the smallest amount, the correlation is still quite high. And you don't gain a lot by using a whole section of tissue, which you would see there on the far right at 167 millimeters squared. But in general, what we see is good correlation across all 4 samples using very small amounts of tissue. So here's another way to kind of look at the data and see how these samples cluster together, or if they do. And for the most part, the colon samples using different amounts of tissue all segregate together, with the exception of a couple of the samples where the 1X sample did not cluster with the rest of the smaller amounts of tissue. And there we might think about one of the possibilities is the heterogeneity, even within a slide, might be compromising the clustering and the similarity of the samples. So there I'm going to conclude or kind of summarize what we found. So the advantages, obviously, of the HTG technology is no extraction is required. Now, I am aware that nCounter does have a facility to also not do RNA extraction, and we just really didn't have the time to make that comparison. But, and so the data that we used was with RNA extraction. The input amount was 1 section in the case of HTG Edge and 2 sections for NanoString, although we're quite confident that we can also use 1 section for NanoString. We were able to interrogate 2,560 genes with Edge and only 770. We go up to 800 with nCounter. So the batch size, we can do 8 to 96 samples, 24 samples at a time, pretty similar with NanoString. In both cases, there are 3 days turnaround time. The platform price is better for Edge, but that assumes that you already have NGS capabilities in your laboratory. The hands-on time actually is pretty similar. The cost per sample is here the comparison. I have 2 off-the-shelf prices because the cost per sample for EdgeSeq is also a off-the-shelf, non-custom. The cost per gene is of course lower because you can do more genes with EdgeSeq, and the detection is kind of straightforward reads and ion, and detection in NanoString is a little bit different. So my conclusion is that EdgeSeq eliminates the need for RNA extraction. It works well with ion torrent. It has good reproducibility, good concordance with gold standard, including TaqMan and nCounter, in which you have to do RNA extraction, or that's what we used in this case to make the comparison. There are similar concordances between EdgeSeq and TaqMan or nCounter to what you see between TaqMan and nCounter, both gold standards. And we feel that you can use down to 1 5-micron section with an area of 3.1 millimeters squared. So just to highlight some of the people who worked on this, particularly the work of Corey Lipchuk, who did the molecular profiling. Patrick Gavin did all the analysis, the matrices, and of course, without a pathologist, tissue bank manager, or histologist, we're completely lost. So, and David Korolenko was the first person to kind of introduce this technology to us, and BJ Karen and Fouad Janat have been instrumental in helping carry out this project. And thank you very much.
Thank you for your presentation, Catherine and John. It is now time for the Q&A. This is your opportunity to ask Catherine and John your questions. To ask a question, all you need to do is press the Ask a Question button on your console, type your question, and press Send. So let's look at our first question, and it asks, what's the resolution threshold for HTG EdgeSeq? Is that a question you can answer, perhaps, John?
Yes, was the question regarding the number of cells in the FFPE sections?
Yeah, that's right. So the other part of the question was how does the FFP section area compare to the cell numbers?
I think Dr. Guy might be better served answering that question.
Sure. Catherine.
So I guess we, in general, look for cellularity, but precise number of cells, I can't tell you, but it's pretty similar depending if you're familiar at all with, if you look at the surface area and the number of cells, I think it can be computed. Honestly, I don't know that. So probably a better guideline for right now is the pictures that I showed you, although the resolution may not easily compute to the number of cells. But so I actually don't have that information.
Sure.
Yeah, and we can also, when we validate our assays, we look at about 1,000 cells as being what our comfortable or limit of cellular input as well.
Excellent. Thank you both. And our next question asks, what are some of the applications the Oncology Biomarker panel has been used for? Is that something you can help with, John?
Yeah, certainly. So we really, when we designed the panel, we designed it to try to be a kind of a jack of all trades. It's certainly been used to distill larger signatures that were developed on RNA-seq or other microarray panels, which are not necessarily deployable or the most easy to run technologies, then try to simplify them so they can both validate and potentially deploy that signature in a more in a more process-oriented setting. It also has been used for biomarker discovery. As mentioned, there's 2,600 total genes on there, so it has a wide range of view on different aspects of the oncology transcriptome.
Excellent. Thanks, John. And our next question asks, how are you able to measure RNA without RNA extraction?
Yeah, I'll take that question. So it really has to do with the differences in our technology. As mentioned in the presentation, it's really quite elegantly simple. We just need to have the DNA probe hybridized to an RNA molecule. So that RNA molecule can be in solution, as is traditional with extracted RNA, but it can also be cross-linked to FFPE material, and when whatever that cellular debris is that you have in the bottom of your tube when you actually lyse and rehydrate those samples. Just because of the ability, and we don't need to have an enzymatic step, polymerase step that amplifies that nucleic acid, we can get by with just the hybridization of the nucleic acid and the nuclease attacking that particular probe.
Excellent. Thanks, John. And another question that's come in, I'm not too sure whether you can, either of you can answer. And it asks, how does the Wingman probe protect the qPNA probe? And how is the stability guaranteed during the S1 nuclease digestion? Can either of you help with that question?
Yeah, I can address that. We use a combination of temperature and conditions to ensure that those wingmen stay, so that portion of the probe stays double-stranded during that stage. It was part of our thorough testing of the technology and the chemistry before we deployed it. So it's really just just the solution conditions and the temperatures we use.
Excellent. Thanks, John. And our next question asks, are you able to measure long-read non-coding RNAs?
We have done some pilot work on these. It's certainly, from a feasibility standpoint, completely feasible. It's something that we're looking at developing products around in the future.
Excellent. Thanks, John. And just a quick reminder to our live audience, you still have some time to ask a question. Remember, all you need to do is press the Ask a Question button on your console, type in your question, and then press Send. And our next question asks, how long does it take to run the HTG EdgeSeq assay?
John, are you answering that question, or?
Go ahead. Go ahead, Dr. Gile, you can take that, please.
So the limitation, so the first step is really, in terms of making the EDGE product, is really quick, meaning depending on how many samples you are and how long it takes you to scrape your sample into the lysate tube. So that part's really quick. And then the machine takes several hours. John, like 4 hours or something, right?
So the machine actually takes 20 hours. It's an overnight processing step.
Hours. So that's an overnight process. And then the next part of it is, depends how long you have to make a library. So you have to, depending on what your process is for making the library and doing the sequencing, is going to determine the length of time you need for that next step. And that's going to vary with your technology.
Yeah, our experience with this is, it depends on how operationalized you make the process. But if you're in a lab with working SOPs, typically in a one-shift laboratory, you can turn samples from tissue on the slide to results in hand in about 48 hours. If you're working in a two-shift laboratory, you can get that down to about 36, 38 hours. If you have operationalized it and removed any of the attritions that you have for time.
Excellent, thank you both. And our next question asks, what is the dynamic range of the assay?
Reasonable question. I think it was to four orders of magnitude.
Yeah, in our experience, it really depends on how many sequencing reads that you put on our assays and how many reads you allocate per sample. From a theoretical standpoint, we're operating at about the same level as the sequencers themselves. So we routinely see 4 log 10 range. You can see 5 log 10 range if that level of expression difference actually exists between the genes. It's actually fairly rare in the mRNA world to get that, but that can be captured if it exists.
Excellent, thank you both. And our next question asks, which sequencers can I use with the assays? John?
Yeah, so we currently support both the Illumina and the Ion Torrent family of sequencers. We validate assays and the probe adapters for both of those. Other sequencers that are emerging, we certainly have our eyes on those, but we currently do not provide commercial products for them.
Sure, thanks, John. And our next question asks, can you measure splice variants?
The short answer is yes, we can. We've certainly done feasibility on that in specific assays. Our standard assays, the Oncology Biomarker panel and the IO panel that we discussed briefly today, are not set up to do that. But if we do create a custom panel, we can certainly set those up.
Excellent, thanks, John. And our next question asks, assuming that the standard assuming that standard QNPA reaction is carried out after the recovery of tissue from slides, is it possible to carry out QNPA chemistry on the slides? Hopefully you managed to get the question from that. John,
I did. Yeah, so we've been asked this question. It's not something that we're typically working on the slide itself. That would involve a whole series of different potential ways of actually detecting the differences going on in between the cells. In theory, it's an intriguing question. In theory, it might be possible, but I think at the end of the day, it's probably not something we're going to pursue immediately.
Thanks, John. And our next question asks, how many samples per run or per number of counts per sample?
Yeah, this really depends on the number of genes that we're looking at in a particular assay. So if we're looking at a small assay of 100 or a few hundred genes, we can easily run, we're talking about a few 100,000 reads per sample. For our larger panels, we recommend 2 million reads per sample for the OBP. So it's really a function primarily of the number of genes we're looking at.
Excellent, thanks, John. And our next question asks, is your chemistry able to detect mutations in DNA like SNPs?
That's not something we have currently as a commercial product, but keep your eyes on us. That's something that we are in developing at this time. We're very excited about.
Excellent, thanks, John. And our next question asks, does the age of the FFPE clock impact the reactions?
The short answer is no. To our to our knowledge of our testing, we have not. We recently tested and built a CE-IVD product on samples that were ranging from over 15 years old all the way up to 2 years old, and got very similar results on those particular samples. So we do not need to have fresh sections to test these. There does not appear to be any significant impact of aging of the blocks or the cut slides.
Excellent. Thanks, John. And I think our last question asks, and hopefully this is a very simple question for you both to answer, as to whether the slides will be available after the webinar. Is that something that we can produce for them?
Actually, no, I don't think so. But I could talk to any individual if that's useful.
Brilliant. I'm sure we can pass on their details after the webinar.
Okay.
Brilliant. And our next question, which is from Saga, asks, is a question with regards to input controls. Their study is aiming to analyse changes in gene expression in response to anti-cancer drug treatment. And from their pilot study, they know that a drug X causes changes in gene expression and apoptosis in cancer cells. How would control variability in the input for the RNA expression analysis in HTG oncology biomarker panel techniques? Is that something you guys can help out with, perhaps?
As I understand the question, it sounds like how do you? I mean, this really ends up being we would put in a similar number of cells as we can at the beginning.
Sure.
Obviously, the drug might create some apoptosis or some other effects on that and kill off some of the cells. But we could also obviously just do data level normalization on those results. We typically use a global normalization scheme, but there's also housekeeping genes if that is more suited to the purpose. It really depends. You have the options about how to normalize that input.
Thanks, John. Well, I'm afraid that's all we have time for today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. I would like to thank Dr. Catherine Poe-Guile and John Lucky for their presentations and for answering your questions. I would also like to thank the webcast sponsors, HTG Molecular Diagnostics, and of course you, the audience, for taking the time to be with us today. Thanks for watching, and I hope you'll join us again for our next webcast.